আজকের প্রোগ্রাম হলো যে প্রথমে introduction, meet the professor. I will introduce myself, briefly, and I will actually show you my journey, how I started, where I was born, and how I actually started my career for science. And the second one, I will be talking about motivation from Big Bang to protein and metabolites or metabolomics. And then we will start the main actually lecture. Today we'll cover mass spectrometry instrumentation. Mostly we'll be talking about all the component of mass spec, and also we'll cover some of the history. Very basic thing about mass spectrometry and different type of mass spectrometry, and then we'll take 10 to 15 minutes break, and then then we will learn how we can actually analyze mass spectrum, especially protein and small molecule mass spectrum, and then we will also show how you can download some of the key software we'll be using in this training program. Any questions so far? Therefore, English check. Okay, so is there any problem if I actually speak in English? Is there any international international applicant or you can speak English? Okay, because I'm a little bit more comfortable because my Bangla is kind of broken. Okay, so I will try to mix sometimes. Okay, and you are allowed to ask me question in Bengali. Okay, that's not a problem. Thank you.
Sir, I have a request.
Yes.
Sir, can you record the class, please, sir?
Yes.
Okay, thank you, sir.
Recording in progress.
Okay, so as I mentioned that at the very beginning, I would like to introduce myself. Okay, as you know, that I'm also from Bangladesh. So I was born in actually a remote village, which is called Anantapur. Anantapur means this is the last village actually. After Anantapur, there is no village of Bangladesh. So then we have Indian border. Okay, and it's very remote. So I was born there, and I started my elementary or primary school there in my village. When I was born there was no electricity in my village, so it was very remote and isolated, and very far from big city like Kurigram and Rangpur. Actually, that time Rangpur was mostly famous city. Okay, and then I moved with my father in Khulna. So I started my primary school also in Khulna. So I studied in Shiromani primary or elementary school up to grade fifth grade, and then I moved to Cantonment Public School and College in Jahanabad, like in Khulna. So I studied there up to seventh grade, and then I moved actually to Srimangal with my father because he retired from his army service, and then he started his own business in Srimangal. which is Sylhet. So I see my childhood as very delocalized. Okay, so from started from Anantapur, then Pune, and then Sylhet Srimangul. So I actually matriculated. I completed my SSC from Srimangul, and then I started my college in Dhaka City College, and then I went to Jahangirnagar University. So when I was in college, I started loving chemistry because of my college teacher. He was super good in chemistry. Then I went to Jahangirnagar. My major was in chemistry, although I also minor in physics and mathematics. Okay. So when I was in Jahangirnagar, I met with this renowned professor. Maybe some of you know or may not know him. So he is Syed Safiullah. He is one of the actually famous and scientist as well as polymath. Okay, polymath means he can teach you philosophy, chemistry, biology, history, mathematics. So he is one of the polymath. Many people maybe don't know him, but he is one of the um, uh, genius professor in Jahangirnagar University and all over Bangladesh as well. Okay, so I started working with him to make some filter material that can remove arsenic, because as you know that we have arsenic contamination in our groundwater. So my initial research was to collect water from different tube oil and then measure how much arsenic we have. So detection is kind of environmental analytical chemistry. And then we also created some filter materials from natural source, try to remove arsenic from water. Then I finished my honors and masters from Jahangirnagar. I briefly started teaching at Dhaka City College. And in 2009, I actually went to Newfoundland, so Newfoundland in Canada. So you know, there's Irish people. So these people living in Ireland, they are called Irish people. So they're very good actually fishermen, and they started actually fishing nearby land of Ireland. But some young fishermen, they want to actually explore. They want to actually go meet Atlantic. But the old people reminded them that okay, if you go, you may be lost. You may not be able to return your homeland again. But they didn't listen. You know, young people, they do not listen. That way they invent and explore a lot of things. So they started their journey, and they're getting a lot of cod fish and Atlantic salmon, and they were very happy. And then they thought that okay, let's move further. And that way actually they lost themselves, and they were not getting or they are not seeing any land for a couple of months. And then around 1400 they actually found fast land. So that time they were not speaking English, so they were speaking kind of French, Scottish language, and they named this island Terra Nova. Terra means land, Nova means new, so Terra Nova. Then when British came, as you know, the British went all over the world, they gave name Newfoundland. Okay, so this is the first actually European explorer. They first actually found this land, that piece of land. So I was there. There is a university, Memorial University of Newfoundland and Labrador. So they have a port city. St. John's is very beautiful city, but only one problem in Canada is that you will get a lot of snow. Even in October, when fall just started, you will get snow. So this is my first snow in 2009 when I went there. Uh, so this is in actually in I think early September or October. So I started my research work with Professor Raymond Poirier. So he's a Canadian, French Canadian, um, and I started working with him on computational chemistry, very different chemistry that I used to do in Bangladesh, from environment to computational chemistry. That's why I actually founded a research center first with computational. Then now I'm moving to proteomics and metabolomics and mass spectrometry techniques. So I work with him. Then after finishing my master's in Canada, then I actually moved to France with three French words. Bonjour means hello, comment ça va means how are you, and merci, because the French people, they do not like English, so they prefer their language so much. So at least you need to start conversation in French, okay? And then you can say that's bonjour, je n'ai pas parlé français, je parle français un petit peu, excuse-moi. Then you start talking in English. They know English, but they do not prefer English for their fast communication. So I was working in Philippe Dugo Group in CNRS. CNRS is very similar institute like Atomic Energy or BCSI like Bangladesh. So it's a government institute. It has headquarters in Paris, but it also has other center around the France. There are 6-7 big CNRS centers. So I was working in Lyon. That's the second largest city in France. And then I was working with Philippe Dugo, so I gave him a nickname, Napoleon, because he is only 5'4", but he was very, very intelligent and smart. So that's why I gave him a nickname, Napoleon. After finishing my PhD in France, then I joined French Atomic Energy as a research scientist there, and there I also work with two other scientists, Christophe and Sebastians, and where we actually developed a new type of mass spectrometry, and then we actually also published this work in Science. So now I actually want to actually share what type of work I did. So during my PhD, so I try to break down the protein using laser. So usually biologists, they use different enzymes to break down protein into peptide or amino acid. So, but my supervisor is a physicist, so he loves laser, and he wants to use laser to actually break down the protein into amino acids. Okay, so we use ND Young laser, and then we generate fourth harmony. 230 nanometer wavelength, like this is UV light we generated from this laser, and then we also use CO2 laser or IR laser, which actually give us 10.6 micron wavelength, which actually gives us very low energy, and UV is very high energy photon. So UV light can break down the carbon-carbon bond inside the protein, and IR laser can actually break down the carbon-nitrogen bonds inside the protein. So then we couple UV and IR together with the mass spec. So this was our mass spec, and then we try to see how laser actually cut the protein into small pieces. Okay, we use a model protein, ubiquitin, small protein 76, and then we try to see how it's actually fragment. Okay, so this is the sequence of the ubiquitin. Sequence means all the amino acid present inside the ubiquitin, and when we actually use IR laser or CO2 laser, we see that it mostly breaks the carbon-nitrogen bond, so which is actually BY. And but when we use UV laser, it actually breaks carbon-carbon bonds. Also, so there are three types of bonds inside the protein. So carbon-carbon, carbon-nitrogen, and nitrogen-carbon. So when we actually couple both of the laser together, we see that actually we get more fragmentation. That was our hypothesis that both laser actually can cut the protein into pieces. That's actually published as a cover article in Journal of American Society of Mass Spectrometry. And then when I moved to France Atomic Energy, so that there I actually developed a new type of mass spectrometer. So we created a balance beam. Okay, so this is called balance beam, where you can actually jump the okay, so those who play this sports, gymnasist or something, so they actually when they jump on this balance beam is vibrate. From the vibration we can actually sense their weight. Okay, so same thing, we created this balance beam inside the silicon chip. Okay, and then we deposited particle, and when we deposited particle, so this is the initial frequency, this is the Hooke's law, maybe you studied in high school physics. So you see this is spring constant and this is the mass, and then when you deposited something on this balance beam,
কি
you
অবস্থা
see the
ভাইয়া?
frequency shift. So when you see frequency shift, if you have bigger substance, bigger molecules, you'll see more shift. If you have small molecules, you'll see small shift. From there, you can actually calculate this mass. Okay? But this is from physics, but then we need to build the instrument. So we build a new instrument where we use this nano electromechanical system that's called NEMS, which we use as a detector. So we generated the gaseous ions or gaseous molecules, and then we transferred through aerodynamics. I will be talking this maybe later on. You do not need to understand all those things right now, but I'm just sharing things. So you generated gaseous ions, and then you transfer these ions to the detector, and but this is the real instrument that's actually we developed in France Atomic Energy. We use this machine to measure the mass of virus capsid, okay, viral capsid. So we measure the mass of filled capsid as well as empty capsid, okay. Empty capsid or bacteriophage. So filled capsid means inside the capsid you have DNA or genome, but empty means you do not have DNA. So if there is no DNA inside the capsid, so the mass is, the theoretical mass is kind of 26 megadalton. But with genome, the mass is around 105 megadalton. Okay, 1 dalton means gram per mole. So megadalton means 10 to the power 6. Okay. So using our instrument, we measured the mass of empty capsid is 27.2 megadalton, which is very close to 26. And using this same machine, we measured the mass of filled capsid, which is 108, which is close to 105. Okay, so that's what we published in Science. Then again, as I mentioned, as I love actually delocalization, moving from one place to another. So then I actually moved from France to USA in 2018 after finishing my job in France Atomic Energy, and I moved to Pittsburgh, Pennsylvania. Pittsburgh is a steel city where actually all steel are produced. There are a lot of industry or steel factory there that was actually established in mid 1800. Still there are some. So there is a university called Carnegie Mellon University at Pittsburgh. Is a very famous university. So I started working with another professor. His name is Mark Baer. So there I also started working on developing a new type of mass spectrometry, a new type of detector that's called superconducting tunnel junction. So this year Nobel Prize actually got to superconducting tunnel junction physics. Okay, so this is a detector that we put in cryogenic condition, means where the temperature is very very low, zero point 0.3 Kelvin, using liquid helium, and then we actually generated protein. We trap this protein and then we shoot this protein on the detector. So good thing about this detector can actually, when we put it in the cryogenic condition, it can actually detect lot of things that traditional mass spec cannot detect. Also, we want to measure the energy of the protein. So protein sometimes they are folded, they are sad, they are folded. Sometimes protein get excited. Okay, when they get excited, they are unfolded. Okay, so using this detector, we can measure the energy of folded protein and unfolded protein, and also we can increase the detection level because in cryogenic condition. So we found lot of different protein like monomer, dimer, and trimer, tetramer is kind of one of plenty, and we also measure the energy. So traditional mass spec can measure the abundance, but this new detector that can actually measure the energy as well. So we published this article in Analytical Chemistry. But this was the instrument that I work in Carnegie Mellon University because it's a new built, home built or custom built instrument. There was no actually instrument available that can do. So we built it by taking different parts from different leftover mass spectrometer. We put them together. We write down the code to drive all the voltage inside the mass spec, and then it was very difficult to work with this type of instruments. Then in 2021, I moved here in Kennesaw State University as a assistant professor. Hopefully next year I will be promoted to associate professor. Here I do a lot of things. I do not have traditional
ভাই
boundaries
কি অবস্থা?
between
বউ
chemistry,
ফোন দিছিল নাকি?
physics, and biology. Okay,
বউ
so
ভিতরে আইছে?
so I started doing peptide therapeutics.
যাক আলহামদুলিল্লাহ এটাই তো ভালো।
I'm also doing native
কবে
mass
আসতেছেন?
spectrometry, proteomics, lipidomics, metabolomics,
যাক
also some
আলহামদুলিল্লাহ তাই তো আপনি তো
ambient
লালে
ionization
লাল এখন।
technique, and of course
বউ
I'm also
পাচ্ছেন,
doing a lot of computational study.
পার্মানেন্ট
But
চাকরি
now
আর
I
কি
do
লাগে?
not do a lot of computational
ওখানে তাহলে
chemistry
ভালো
here,
মানে এই বাসায়
as I
থাকবেন নাকি অন্য
founded a
বাসায়
research
যাবেন? অন্য
center in
বাসায় যাবেন?
Bangladesh, so mostly they are doing. I'm just supervising them, uh, um, so that they can
আচ্ছা
do
আচ্ছা।
actually
যেহেতু
good
বউ আসতেছে
computational
মানে
chemistry
একটু
there.
একটু
I have
একটু
a big
বড়
group
বাসা
here, so I
তাহলে
love
ভালো
it,
হতো।
working with the student. Each and every semester I have kind of 30-35 students working in my lab.
হুম
Okay,
হ্যাঁ
so this is
রিমোট
the,
এরিয়া করলে সাধারণত আর কি।
I think it's a very detailed introduction about me. I hope it's not boring.
আচ্ছা আচ্ছা।
Recording stop. Is there any question?
হ্যাঁ 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 হ্যাঁ।
If not, then we can move
আচ্ছা আচ্ছা।
to. I have a question. It's live here. You told that you used
হ্যাঁ হ্যাঁ
you waited the
হ্যাঁ হ্যাঁ
bacteriophages. Yes, in France. So did you I can say that the felt the vibration that bacteriophage produces, and then you just waited it,
আচ্ছা আচ্ছা
wait it, yeah. So actually, we did synthetic biology. So the capsid protein or the related protein
হুম
that make the capsid. So first we express
ভাঙানো
and
কম
purify those protein, and then we put in a system that's actually
আচ্ছা আচ্ছা
synthetically we made these. It's not that we
ও আচ্ছা
isolated
ওখানে কি
this
মানে
capsid from
কাজ
virus.
পাবেন পাবেন পাওয়া
We
যাবে
actually artificially made this capsid. Okay, so like
মানে
synthetically.
আসা যাওয়া
Synthetically
কিরকম
means
টাইম
you just
লাগবে
need to put all the ingredients of related all these
হ্যাঁ
capsid protein and coat protein, and then they actually
হ্যাঁ হ্যাঁ
spontaneously they make the capsid.
ও তাই তো
And then
বিশাল
in another experiment we added the
কাছে
genome, and then genome will be enter referring to
ভালো।
the capsid. So it's very spontaneous. We collaborated with a synthetic biology group in
ভালোই আর কি।
France. So they actually made this empty and filled capsid for us. And then once we get this capsid, then we prepared our sample, and then we transfer this capsid from
না ওয়াইফকে কি
liquid to
সি3
gas
ভিসা দেয়?
phase, and then
সি3
we
কি টুরিস্ট ভিসা
actually
নাকি এটা কি ইয়ে ভিসা
try to
সি3?
transmit them to the
ওয়াইফকে কি
detector,
টুরিস্ট ভিসা
and
দেয়?
then using that NEMS detector. That is
ও
a very
আচ্ছা
new
আচ্ছা।
detector actually.
আমি এখানে আমি এক ভাইকে জিজ্ঞাসা
There is
করছিলাম
no
উনি
machine
বলল যে
available.
ওয়াইফকে
Actually, there
তো
is no company
ওয়াইফকে টুরিস্ট ভিসা
they
দিয়ে
produce
দাঁড়ান
this
করলে
type of
জিজ্ঞাসা করবে আপনার তো
mass spec yet.
হাজবেন্ড থাকে আপনি
But
টুরিস্ট ভিসা
hopefully
যাবেন
in
কেন
10-20
আপনি তো
years
এমনি
we can
স্পাউস
purchase
ভিসা
this
যদি অ্যাপ্লাই
NEMS
করতে পারেন এই
MS.
ধরনের কোশ্চেন নাকি জিজ্ঞাসা করে আমার বন্ধু এই জন্য আমি একটু
Okay,
ডিমোটিভেটেড
then
হলাম আর
what
কি
you mean
কি
is
বলবেন
synthetic means you just made it in the lab, the
হ্যাঁ হ্যাঁ
bacteria.
Yes, yes, yes. We made this capsid in the not us, our collaborator actually.
Okay, okay, so thank you.
হুম
Any other question?
হুম
As I mentioned that you can ask me question. তোমরা আমাকে
রিজনটা কি
প্রশ্ন
আপনার
করতে পারো বাংলায়, কোনো সমস্যা নেই। আমি বাংলায়ও উত্তর দিতে পারি আর কি, ঠিক আছে? আমি জানি যে
আচ্ছা
একটু
আচ্ছা
ইংলিশে কথা বললে একটু প্রথমে একটু হেজিটেশন হয়। হ্যাঁ,
আপনি ব্যালেন্সের কথা বলেছিলেন আসলে ওই
হুম
ব্যালেন্সে
হুম
কিভাবে ফাংশন করছিল এটা আসলে এখানে কিন্তু বুঝি নাই।
ওকে, সো এটা আসলে জাস্ট একটা ইন্ট্রোডাকশন। আমি আমার ওই সায়েন্স পেপারটা পরে শেয়ার করতে পারি,
আচ্ছা
যেটা
আচ্ছা
তোমরা পড়তে পারো আর কি, ঠিক আছে? তবে আফটার এই ক্লাস করার পরে
ও
তখন
আচ্ছা
তোমরা
আচ্ছা
অনেক
মানে
জিনিস
এটা কি টুরিস্ট ভিসা কি
বুঝবা
আবার
আর কি, ঠিক
পরের বার
আছে?
আবার টুরিস্ট ভিসার জন্য অ্যাপ্লাই করছিল তখন দিছে টুরিস্ট
এরকম
ভিসা
না যে প্রথমেই সবকিছু বুঝে ফেলবা,
হ্যাঁ হ্যাঁ হ্যাঁ
ঠিক আছে? আহ, তো এই
আচ্ছা
ক্লাসটা
আচ্ছা
করার
আচ্ছা
পরে
কিন্তু
যে
ফ্যামিলি ভিসা
বিভিন্ন
না দিয়ে
ধরনের
টুরিস্ট ভিসা ধরে
ডিটেক্টর
নিছে আর
কিভাবে
কি
কাজ করে সেই
ও
জিনিসগুলো ক্লিয়ার হবে।
হুম
Yes.
আচ্ছা
কে
আচ্ছা
যেন
আচ্ছা
কোশ্চেন করতে চাইছিল?
তাহলে আপনার বউ তখন
That
কি
as
আপনার
to
উনি কি
that
তাহলে টুরিস্ট
I
ভিসার
saw
জন্য অ্যাপ্লাই
on the
করছিল
detector
ডাইরেক্ট
there are so
প্রথমেই
many energy levels, like 60 cal. Is there? I forgot.
Okay,
ও
so
আচ্ছা
that's
আচ্ছা এখানে
energy
কি
level
আপনার স্যালারি
means
দেখানো
actually
লাগে
that
টুরিস্ট ভিসায়
you're
বা
talking
টাকা
about the
দেখানো লাগে
laser, I
নাকি কি
guess.
দেখাইতে হয়
Well, it was in the gas chamber.
হ্যাঁ হ্যাঁ
So my question is, why there are so many energy levels?
হ্যাঁ
Because
হ্যাঁ হ্যাঁ
is it because when your particle pass
আচ্ছা
over
আচ্ছা
the
মানে এখন হলো
gas
ইলেকট্রন
chamber,
প্রোটন
they
দেখেন
unfold
ঠিক
more
করা
and
যায়
more,
এটা
and then
অনার্স চেক করা
um,
যায়
every possible unfold time they just capture the
আচ্ছা আচ্ছা সমস্যা নেই আচ্ছা
unfolded protein by
Oh,
laser.
আচ্ছা
okay,
ঠিক
okay.
আছে
You're talking about the STJ detector, okay? Is that right?
Yeah.
Yeah. So we want to actually make the energy of, okay, so protein actually it doesn't have single structure, okay? So many people crystallography people, they think because they freeze the protein and then they have crystal or crystal structure, so they think that okay, maybe protein has only one structure, but it's not true. Protein has many different structure like us, okay? Sometimes we are sad, sometimes we are happy, sometimes we are in different state, okay? So protein also has in different states. Sometimes they are partially unfolded, sometimes they are fully folded, and we want to see what is
ও
their
বুঝতে পারছি
energy
ভালো আর কি ভাই তাহলে আপনি
energy
তাহলে তো 50 লাখ
when they
দেখাইছেন
hit
আর
the
কি আপনি তাহলে আরো
detector,
বেশি
okay?
দেখাইছেন
Uh, um, and when they are folded and when they are unfolded,
হুম
how their energy changes, okay? So the
আচ্ছা আচ্ছা হ্যাঁ হ্যাঁ
traditional mass spec cannot measure the energy.
হ্যাঁ হ্যাঁ হ্যাঁ
You can measure it energy or other experiment, but not with mass spec. So that's why we
স্যালারি কি দেখাইতে
use
টুরিস্ট
that
ভিসা
detector
কম্পালসরি নাকি স্যালারি
that
না
can
দেখাইলে চলে
measure the mass to charge ratio, but also can measure the energy. So that's very new technique.
হ্যাঁ হ্যাঁ
Only two labs actually around the world
আচ্ছা
do
আচ্ছা
that kind of thing. One Switzerland, Zurich, another it was in
ও
Carnegie Mellon.
হ্যাঁ 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 হ্যাঁ
So,
And then
and the reason is because
recently
this
one
one,
lab in Taiwan or
হুম
China, they are trying to build that machine as well. Yes.
ও আচ্ছা 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 আচ্ছা
Sir, I asked a question. Are the different forms
না
in
আমাদের তো
motion
ভালোই
are
লাগছে
they
কি ব্যাপার
isomers
স্যার
or
খাইতে
individual
আর কি
protein has different forms?
Yeah, individual protein. They are not
হ্যাঁ
isomer.
হ্যাঁ ঠিক
Um,
বুঝতে পারছি
so they have different
হ্যাঁ হ্যাঁ
conformation. So they have different conformation. Means they can have folded, they can have
হ্যাঁ হ্যাঁ হ্যাঁ
partially
বুঝতে পারছি
unfolded and fully unfolded. Depends on the environment they are in. Okay.
হ্যাঁ
Yes, thank you, sir.
হ্যাঁ হ্যাঁ
Okay. So now the second we are starting the second lecture, but before that I need
বুঝতে
to
পারছি
start
এটা
the
ভালোই আর কি
recording, I guess. Yeah.
মানে
Recording
পেয়েছিলাম
in
আর কি
progress.
যখন আমরা এদিকে হচ্ছে নেই না আমার হ্যাঁ
Okay. So can you see my slide
আচ্ছা আচ্ছা আচ্ছা
for the second lecture?
Yes, sir. Yes, sir.
Yes.
Okay, so although this is not very related with our class, but so at the beginning I try to motivate you guys, okay? So with something that maybe you never think of, okay? Or maybe you know, but you didn't think of this way, okay?
আমি ভাই এটা
So
ভাই
according to
কুরিয়াতে ভাই
physics,
আমি
that's
মানে
everything started
এতদিন
from
আমি
Big
কইছিলাম
Bang, okay? So
এটা
everything
অনেক
was all
ইনফরমেশন আমি
together
জানতাম না আর কি ইভেন
as a
অনেকেই
very
জানে না আর কি
dense, and
আমি
then we have a
সোলের
bang,
এদিকে এসে আমি হচ্ছে অনেক
Big
ইনফরমেশন আমার
Bang,
হচ্ছে আমি এখন
and
পাচ্ছি
then
আর কি
everything
আচ্ছা আমি আগে জানতাম যে
started,
আপনার হচ্ছে
okay,
মাস্টার্স
according to
মানে
physics.
কোম্পানিগুলোতে
So within few
আপনার
minutes we
ল্যাঙ্গুয়েজ
first
না
got
জানলে
our first
কোম্পানিতে নেয় না
nuclei of
ওরা ডেস্ক
hydrogen,
জব আর
okay?
কি
Then we
আমি
need
এখানে
to
যে
wait
দেখতেছি
for
আপনার
300,000
অনেকে আপনার
years to get the
ফরেনার
first
অনেকে
atom hydrogen.
অনেকে
And
ভিয়েতনাম
then
ফিলিপাইন বা আফ্রিকান আছে
in
অনেকে
few
আপনার
hundred million years
কোনো রকম
we
মাস্টার্স
started
কমপ্লিট করে
getting
ল্যাঙ্গুয়েজ ট্যাঙ্গুয়েজ কিছু জানে না
other
বেসিক অফ
elements like
বায়োলজি
when
আছে
hydrogen and hydrogen they
আমার
fused together, they started forming
ডেস্কটপ বানাতে
helium
বলবেন
and
ওদের
so
ইয়ে
on.
ওদের
So
ওদের
all
আপনার
the
ইয়েগুলো আছে
elements
আপনার একটু সাবজেক্ট ওরিয়েন্টেড
we
জবগুলো
see
আছে
that all the galaxies, stars, and all
ফ্যাক্টর মেইন
expansion
ফ্যাক্টর না
happen.
আমি মানে
So
মানে ইয়ে হয়ে গেলাম
and
যে
then we are
আমি
kind of
এতদিন মানে
presently
কিছু
here.
মানুষ কি বলছে
So we are kind
একটা
of
ঘন্টা
37
কি বলবো আজ গুগল মানে
13.7
গুজব করতে
billion years
যে
old. Okay.
মানে ল্যাঙ্গুয়েজ না
Um,
জানলে নাকি ডেস্কটপ পাওয়া যায়
so
না
this is
আমি
according
এখানে
to
এসে
physics.
এগুলো
Okay.
হচ্ছে
So as I mentioned, that everything started in 13.7 billion years ago. There was a big bang, and then we need to wait for 300,000 years to get first element that's called hydrogen. Okay, and then there is a fusion when hydrogen converted to helium. So where most of the stars are formed during this nuclear fission. So that's why if you look to the sun and all the other star-like sun, they also have more hydrogen and helium. Okay, so kind of 75% are hydrogen in the sun. Okay, and then when the red and red giant star and other star are forming, that time we have nitrogen, carbon, and then when blue star are started forming, then we have sodium, magnesium, aluminum, silicon, phosphorus, all the other, and then when supernova started forming, then we have all the transition metals. Okay, so this is a new periodic table that's actually telling us
আমি
that
এখন মানে
how
আমি ওই
all the
সিভিটি রেডি
elements are
করতেছি আমি
formed
হচ্ছে বিভিন্ন কোম্পানিতে অ্যাপ্লাই করব একটা
during
ওয়েবসাইটে
this
পাইছি
time and
যেমন
from
জব
Big
এন্ড
Bang.
কোরিয়া
Okay.
মানে পিপল এন্ড
So
জব
these
অনেক
are
ওয়েবসাইট ওখানে
atoms.
হচ্ছে
When
ওগুলো কোম্পানি
atoms
হচ্ছে
are
ইউরোপিয়ান
we have
কোম্পানি বা
atoms.
আমেরিকান কোম্পানি
These
ওগুলো
atoms
হচ্ছে
need
আপনার
to join
মানে
together, and then
অ্যাটমিক
they need to
কম্পালসরি
create
না
different type of molecules, water molecules,
ইন্টারন্যাশনাল কোম্পানি
small
ওখানে
molecules, and
অ্যাপ্লাই
then
মানে
they need
করবো
to
আর কি
join
মানে
together.
ওখানে
They
হচ্ছে
need to form
দেখি যদি
amino
হয়
acids,
আমি কিন্তু দুইটা করবো আর কি কোম্পানিতে
and then when amino acid they can join together, they can form protein. These elements can also form like four basic
আরেকটা
biomolecules
বিষয় ভাই
like
হ্যাঁ
protein,
হ্যাঁ আপনার এখানে
carbohydrate,
মানে
lipids,
এখানে
and nucleic acid. Okay,
ভূষণের পোলাপান
and then these
স্টক
molecules they
এখানে
stuck
কোরিয়ান যে
together,
স্টুডেন্টরা আছে ওরা
and
মানে হচ্ছে
they
ফেসবুক মানে
incorporate
অল টাইম
inside the
মানে
cell.
স্টক
We do not
স্টক
know
এক্সচেঞ্জ
how
ওঠানামা
and
করতেছে
when,
বন্ড শেয়ারিং
because it's
এইগুলো না
happened
চালা
over
আলোচনা করে
so
এগুলো
many
আসলে কেন
years.
এটা করে
It's
আমি
very
এই
very
জিনিসটা
difficult
কখনো
to
আপনার
prove
হচ্ছে
in the
ভূষণের কারো
by
ভিতরে কথাটা শুনি
lab
নেই
experiments. So there are two scientists actually in 1952 they tried to
মানে এখানে হচ্ছে
create,
আমরা দেখি সবাই
try to
অনেকেই
understand the
আপনার যারা
origin
আন্ডারগ্রেডে
of
আছে থার্ড
life
ইয়ার করতে পারেন
experiment.
আন্ডারগ্রেডে
So
স্টক
they
মার্কেটে শেয়ার
started
কেনা
they
বিষয় আছে
started putting all the small
আচ্ছা
molecules and
মানে
then they spark
মানে
try
চিন্তা
to
ভাবনা
create
কোন লেভেলের আর কি
earth-like environment where we have thunderstorms and all those
এই আর কি
chaos,
মানে এদের দিয়ে
and
আলাদা
and then they try to see what type of molecules actually is forming. They
অনেক
found
পারছে
that
ইভেন আপনি
actually,
এখানে
yeah,
যে
eventually
উজবেক
this
স্টুডেন্টরা
water,
আছে
ammonia, they
ওদের কেউ
actually
কামলা দেয় না
turned into
কিন্তু
some
আবার
kind
টিউশন
of
শুন ওরা
amino
টাকা দিয়ে
acids
করতেছে
or
এখানে
they
ওরা প্রজেক্ট
form
করে হচ্ছে আপনার ওই যে
amide bonds, okay? But this is
অনলাইনে
the only one
ইয়ে করে কাজ করে
experiment,
আর কি
or
পেইড
couple of
অনলাইনে পেইড
experiments
জব করে
actually done in
ওই যে ওই আকো আর
1952.
ফাইবার
But
আছে
most
না
of our
ওই
current
হ্যাঁ
research
এগুলো
doesn't
করে হচ্ছে
actually
জব করে
support
ঘরে
to
বসে বসে জব
explore the
ফ্রিল্যান্সিং
origin of
ফ্রিল্যান্সিং
life,
এই
how
যে মনে আছে
everything's happened.
ফ্রিল্যান্সিং
So
সবাই
we do
সবাই
not
ফ্রিল্যান্সিং
know
করে
because
মাসে
we
হচ্ছে
cannot
আপনার কত
actually
20,000 ডলার $25,000
produce this
30,000 ডলার
evidence in a lab setting because we cannot
মাসে
create
আপনি
that
ফ্রিল্যান্সিং করে
early Earth-like environment. Okay.
30,000 মানে
So there are a lot of
মিনিমাম
questions. So
20,000 ডলার
as I
আমার
mentioned,
একজনের সাথে আমার
the
মানে ট্রাই
Smith-Muller
করা হয়
experiment.
ইউনিভার্সিটিতে
So
দেখা
from
কথা
there
বলে
they found
প্রোগ্রামিং টোগ্রামিং পারে
different
শিখে
products.
ওরা হচ্ছে এখানে হচ্ছে
Um,
ওরা এখানে কিন্তু আপনার
so
কোনো
and
ইয়ে সাবজেক্টে
different
পড়ায় আর্টস বা কমার্সের সাবজেক্টে
amount
পড়ায়
of
ওরা হচ্ছে
different
এখানে পরে
products that
সব হচ্ছে
form
আর্টিফিশিয়াল
from
ইন্টেলিজেন্সে
their
পড়ে বা গ্লোবাল
experiment.
কনভার্জেন্সে পড়ে আর কি
So then
আর ওরা হচ্ছে
you know
এটা
if
একটু
you
পড়াশোনা করে সাইডে ওরা হচ্ছে এই বিজনেস এবং
visit
এইসব কাজ
Vatican,
করে
you will
আপওয়ার্ক
see the
ফাইবার
Sistine
এগুলো হচ্ছে ওরা
Chapel.
হচ্ছে মানে কাজ করে
This is
ওদের
the
হচ্ছে সবগুলো ওদের
masterpiece of
কাস্টমার হচ্ছে
Michelangelo where
কানাডা
he
আমেরিকা
actually
বা ইউরোপের আর
depicted
কি
Adam
ওরা হচ্ছে
and
কাজ করে
this
দেয়
old
তো
guy
কাজ করে
depicted as
টাকা
a
পাঠায়
god, and this is Eve, and this is
আমি ভাই এখানে কোনো
all
কামলার
of
আমি কোনো
us.
ইয়ে
Okay.
দেখিনি আর কি
This means we are
হ্যাঁ
coming over time, and we also have a not very
হ্যাঁ
you know stable relationship between man
ডোর
and
খুলে দেয় হ্যাঁ হ্যাঁ
God.
হ্যাঁ
Okay.
আচ্ছা মানে
So it
আমি
seems like
এখানে
after
আড়াই ডাবিশ করছিলাম
Adam or the
আমি
early
এদের বলতে দেখুন আড়াই ডাবিশ বাইক খুঁজে
they
পাইলাম
were
না
born
আছে হয়তো বা
and they started
অন্য কোথাও আছে
living in
কিন্তু আমি
cave.
আমার
Okay,
চোখে
so
আমি
we
কারণে
were
আড়াইটা
kind
অবশ্যই
of
যাওয়া
in the
সম্ভব
cave,
দেখি
and then actually the cave ancestor, or I usually call them cave chemists. So they actually first invented whatever they were getting. They were just rubbing
আচ্ছা আচ্ছা
together, and they are
হ্যাঁ
expecting
হ্যাঁ হ্যাঁ
some miracle will happen, and eventually miracle happened. They actually invented the first chemical reaction, which is called fire. So when
আচ্ছা
first
আপনার তো
chemical reaction was invented, then they did other experiment. Okay, so
হ্যাঁ
they
আপনি
now they have
এরিয়াতে
they
থাকলে
know how
কোম্পানি
to
ভালো একটা পজিশনে
create
যাইতে পারলে
fire,
ভালো
how
হয়
to make fire, and then lot of
আপনি হ্যাঁ
other people came. People they came try to learn
আপনি ভাই
you know
ডেস্কটপের জন্য
how to
ইয়ে
make
করেন
fire. Okay, so there
অনেকগুলো
maybe
একটা
there was
ওয়েবসাইট
a
আছে
school.
এবং আপনি
So
ওয়েবসাইটে ভালোভাবে খুঁজেন
then
না আপনি
they
কিন্তু পাবেন যেটা
teach
আপনি ল্যাঙ্গুয়েজ
these
পাবেন
things to
আপনার
other
কোরিয়ান মাস্টার্স ডিগ্রি আছে
peoples and other
আপনি
people,
যদি
and they
ডেস্কটপ
share
ভালোভাবে খুঁজেন সিভি
how
যদি
to
ভালো মতো
create
ইয়ে
fire.
করেন
And then some
ড্রপ আউট
other
করেন
people
মেক
started
করেন
doing
সিভি
some other
তারপর আপনি কিন্তু
experiments. Some
একটা
people
ডেস্কটপ
started
পাবেন
putting maybe
যেটা
wood
আপনার
on
ওয়ার্ক
the fire, and when they put
যেটা
wood
ওয়ার্ক
on the
এক্সপ্রেস
fire, they
হচ্ছে
saw that
অলরেডি
fire intensified, and when fire intensified means it gives us comfort in the winter,
এক্সপিরিয়েন্স আপনি
and also all the
খুঁজবেন ভাই
animals
ওয়েবসাইটে
whenever they
খুঁজবেন
see fire
প্রচুর
they
ওয়েবসাইট আছে
kind of run
ওয়েবসাইটে খুঁজলে
away.
আপনি
So
পাবেন
that's
পাচ্ছেন তো
actually
ভাই
give us
অনেকেই পাচ্ছে
things that okay we
খুঁজলে
can
আপনি
use this fire for security or for comfort and lot of things. Then some of the chemists, cave chemists, they started thinking, okay, let's do more experiment. Okay, so let's put
আপনি
you
ওয়েবসাইটটা
know
দেখেন আর কি
animal
দেখে আপনি একটা আইডিয়া পাবেন
on
আপনি
the
তখন
fire.
আপনি
Okay,
নিজে
so
আপনার
they have
এরিয়াটা
maybe
খুঁজে বের
pig,
করতে
or
পারবেন
they maybe have cow, or goat, or bison, or whatever buffalo. They killed it, and
আপনি
then they
মানে
thought
এগুলো
that okay,
শিখেন
let's
তাহলে আপনার
what's
হচ্ছে
happening
ধরেন
if we put
এটা
this
ভালো একটা
animal
মানে
on the
এখানে
fire.
প্রচুর
They
ডেস্কটপ
put
পাওয়া যায়
things in the fire, and they saw and smell there's a beautiful aroma is coming,
হ্যাঁ
and then they started eating, and they find it, oh my god, this is a best way we can use fire to cook something. Okay, and then when you put some wood on the fire, so what's happened, all the cellulose and all the lignin they actually evaporate as a carbon dioxide and water vapor, but all the metals in the wood they actually deposited inside here, in the
আমাদের
ashes.
যেমন
Okay, so
এতদিনে
ash
এখন যেটা
also
স্কিম
means
হচ্ছে ভাই,
actually
কোরিয়াতে আসলে আপনার জবের জন্য
metal.
পারফেক্ট
Okay,
আছে,
so
ইন্ডাস্ট্রি
ash contain a
কোম্পানিগুলোর
lot
জন্য
of
কোরিয়া
metals,
ভালো আর
mostly
কি।
sodium and
পড়াশোনার জন্য
potassium.
ভাই কোরিয়া আমার কাছে
In the wood
মানে
also have a
পড়াশোনা
lot of
করার
sodium
চেয়ে
and potassium.
মানে
And then the
কোম্পানি জব
fat
করাটা হচ্ছে
actually
ভালো
mixed
আর
with
কি,
these
আমার
ashes
জন্য এখন
and
মনে
it's
হচ্ছে।
actually create a lay or something
মানে
that
কোম্পানি
can
জবে এটা
that
কি আপনার আপনি
people
ভালো একটা
started
কোম্পানিতে ঢুকেন না,
using as a
আপনার হচ্ছে
soap. Okay,
একটা
so this
বেতন
is how
মিনিমাম 30 লাখ
the
এখানে স্যালারি আর কি,
ancient
মানে
soap
কোম্পানিগুলোতে
was
মিনিমাম
made
30
from
লাখ দেয়।
animal fat
আপনার হচ্ছে
when
বোনাস
when
দেয়,
it's mixed
ইভেন
with
কোম্পানি যদি
alkali
ভালো হয়
in the
সে যদি ভাবে
ashes.
আপনার
And then some people they
কোম্পানি
thought
যদি
that
আপনার
okay,
ইয়ারলি
let's
যদি
what's
একটা
happened. They are
ধরেন
craziest
600
people. They thought
মিলিয়ন
let's see what's
ধরেন 100
happening if we
মিলিয়ন
put
হচ্ছে লাভ করলো,
rock on the
100 বিলিয়ন
fire.
100
Okay,
মিলিয়ন লাভ করলো,
so rock
কিন্তু একটা
contain a lot
পারফরমেন্স
of
পাবে না।
minerals
মানে
and
আপনার
then it
ফাইন্যান্সিয়ালি আপনি
contain
অনেক
copper,
মানে
it can
সলভেন্ট হয়ে
contain
যাবেন,
zinc,
প্লাস
it can
আপনি হচ্ছে
contain
আপনার
iron. They
মানে
put
ফ্যামিলি
all the
শিফটিং এর
rocks
জন্য
and
অনেক আপনাকে
they
হেল্প
found
করবে।
figure out that okay,
প্লাস আপনি
the
3
small
ইয়ার্স
fire
জব
cannot
করেন,
help
তো
them.
3 ইয়ার্স
So
এর যে
they
একটা
made a
এক্সপিরিয়েন্স,
big
এক্সপিরিয়েন্স
fire.
দেখায়
They
আপনি
put
হচ্ছে
all the rocks and
ইউরোপ
then the
আমেরিকাতে
rocks
বা
started
কানাডাতে আপনি
melting.
ইজিলি মুভ করতে
And
পারবেন।
then
এই
copper
এক্সপিরিয়েন্স
and
অনেক
zinc
কোরিয়ান
they melt
কোম্পানির
together
এক্সপিরিয়েন্সটা
and
অনেক
created
ভ্যালুয়েবল অন্যান্য কান্ট্রিতে।
brass or bronze, and that's why we have bronzes. Okay, then they started actually improving their chemical method, and they isolated copper from zinc and other metal than copper is, and then they also started isolating iron from other element in the rock. That way they have iron is, and then later they also figure out that if you can put mud on the fire, it turns into very strong solid. Then you put bricks and all those things. Then in the Middle Age, and alchemists, we know all the story. They want to actually convert all other metal, not very expensive, cheap metal into gold. So that was their motivation. They have a lot of stupid ideas. They went to the king and tried to convince them.
বাঙালি অনেকে পাইছে, কিন্তু দুই
They
তিনজন
were
ভাইরে
kind of
পাইছে আর কি,
little bit
তারা
weird
তাদের সাথে আমার
people.
নিয়মিত চা কফি খাওয়া হয় আর কি।
And
তারা অসাধারণ আর কি, বুঝছেন,
they
তারা সোল
started
হচ্ছে
actually
মাস্টার্স পিএইচডি করছে, তো মানে
converting
কোরিয়া সম্পর্কে
these
তাদের
cheap
হচ্ছে চিন্তা ভাবনা
metal
অন্যরকম।
to gold.
এটা আমার ওনাদের সাথে
They
কথা
could
বলার
not,
পরে আমি জিনিসগুলো নতুন
they
নতুন
were not
এক্সপ্লোর করতে
successful, but
পারতেছি।
they created a
এটা
lot
আমার
of
মানে এই
distillation
গত 3-4
and
বছরে
instrument
মানে
still we use in our lab.
মানে এগুলো
Okay?
ধারে কাছে ছিলাম না, বুঝছেন।
And it's continued.
দেখা যাক।
Then German, they were actually
দেশে মনে
influenced
হয়
by
ভাই
these
বিষয়গুলো
alchemists
ভালো
so
লাগতেছে
much.
না,
One of
বুঝছেন।
the
আমি তো
most
ভাই আছে,
important chemist of that
গিয়ে
era
কোন
is
ঝামেলায়
called
পড়বো না।
Hellingberg.
দেখি
So
নির্বাচনটা বার হয়ে
he
যায়।
thought that the alchemists, they are not stupid enough, so they try to convert metal to
Presentation দিয়ে
metal.
ফেলাইছে
So
already.
he wants to actually get
দেখো
gold. He
কেন?
want to make gold out of waste. One example that he want to make gold from urine. Then he started
আমরা
collecting
বউ কবে
urine
আসবে?
from his
মানে কোন
neighbor,
মাসে
and
আসবে?
then of course his wife
Oh,
didn't
আচ্ছা,
like
আচ্ছা।
it, divorced him. So he was
আরেকবার জিনিস
he
আমার
was a
আরেকজন
crazy
ভাই আসবে,
people
উনি
who
হচ্ছে
wants to live with
30
this type
তারিখের
of
দিকে
crazy
আসবে।
people. So, but he didn't stop. Okay, so he collected
উনি
60
আসবে
bucket of
16 আসবে।
urine with his assistants, and then he
উনি
did
জানুয়ারি
a lot
31
of
তারিখে
organic
ফ্লাইট।
chemistry on this urine. Okay, so boil urine to reduce it to thick syrup. So
উনি
you see
আসলে
like
মানে
lot
যাবে
of
হচ্ছে কালকে
organic
24 তারিখ
chemistry,
পরশু দিন যাবে,
and
বুধবার যাবে। তো
eventually he
মানে
saw
রাউন্ড
that
টিকিট
there
করছে
is a
তো,
light coming
ওনার
from his
এই ডিসেম্বরে 24 তারিখ উনি যাবে,
from his
আর জানুয়ারি 30
product,
31 তারিখ উনি ব্যাক
and
করবে।
he thought that is a philosopher's stones, but eventually actually
উনি আমার পাশের
that
রুমে
way
থাকে
he
আর কি।
created phosphorus actually, and then he could not make a lot of money because his recipe was stolen, and then it was stolen by a British, and then he actually sold these things to other people, and then they started actually making matches because they were
সমস্যা
putting
না, আপনার ডাইরেক্ট
phosphorus
ফ্লাইটে
on the
উঠাই নিয়ে
match
আসেন।
so that they
কস্ট বেশি হবে আর কি,
have
অনেক কস্ট হয়ে যাবে ডাইরেক্ট ফ্লাইটে
fire in the red phosphorus that we use in the matches. So that's the whole industry, and this actually motivated other more and more German. There was one of the famous German scientists, Friedrich Hagel. So that time actually a lot of people they believe on vitalism hypothesis. Vitalism is a concept that all the biomolecules cannot be created in lab because nature has this power. Only nature can create this type of molecule. But this Friedrich Wöhler, he opposed those ideas. and he actually put ammonium chloride and silver cyanide together, and then he created a organic or biomolecule that's called urea, because that's urea actually coming from urine. Okay, and urine is whatever the compound inside the urine, they are biomolecules that's made by our body. So then he actually changed the vitalism concept. That's actually, yes, we can actually synthesize biomolecules inside our in the lab, so we do not need biological system. So that's where he invented organic chemistry and organic synthesis, and all the medicine and all the small molecules we see, that's actually started by his idea. Okay. um, so then, for this class, we need to also understand the biology, the chemistry of life process. Okay, so we have DNA. So we have kind of 3.1 billion bases. So far, we know 20 to 25,000 genes, but we still do not know all of their functions. So DNA need to be transcripted to RNA. So then from RNA need to be translated to the protein, and then protein work with different molecules, especially enzyme. They also create a lot of metabolites. Okay. So everything written in the DNA is kind of a cookbook, and then it's a very big book, so you cannot give all the instruction to the ribosome at once. So you need to copy each and every recipe from there. So that's copying is kind of transcription, and then once you give this recipe in a form of messenger RNA, then Shep can see all the codon and then ask is transfer RNA. And then you also need to eat some protein as a food, so that our body can store all the amino acids, sugars, and all other things as the ingredients, and then ribosome can actually make the protein. That's the instruction he received from the DNA. Okay, so we all know this, that we have a phosphate, we have ribose, and then base all together, but it has a nice structure and also has hydrogen bond between the base pair. Okay. And then we have protein. So protein perform various function and malfunction in biological systems. So some protein act as a carrier. It actually transport a lot of things. Small molecule, for example, best example is hemoglobin. So hemoglobin actually all gather in the lungs. Okay. And then they take all the oxygens. It has a container, heme container. So it take oxygen, and then it started transporting oxygen throughout our body, and then it's reached to the each and every tissue where it's actually deliver oxygen. So some protein act as a carrier or transporter. Some protein actually act as a great cook. So for example, the protein present in photosystem 1 and 2. For example, this is a photosystem protein 2. So these are very big protein, and this protein look like pan shape. Okay, so because you cooking, you need light, and you also need a pan. So inside the pan you have lot of coating with chlorophyll. Chlorophyll are coated on this protein, and then it's actually absorb all the light from the sun, get excited, and eventually it cook glucose for all the plants. Okay. Um, and then we have some other protein that's actually are kind of disease maker. So for example, amyloid beta. So there are 3 protein responsible for Alzheimer disease: amyloid beta, alpha-synuclein, and tau protein. Somehow this protein started forming gangs inside the neurons, and these gangs actually get bigger and bigger, and make big gangs, and they usually steal all the beautiful memories we have, and they also erase all those memories we actually store in the neuron, and that's why people become forgetful. Okay, so this is a tau protein. So some protein, they are also kind of disease maker. So to know the protein, we need to know the amino acids. I think we already provided you a assignment, pre-assignment, that you need to know all the amino acids, because this is the heart of the proteomics. Okay. So in the amino acid, you have carboxylic group, amino group, and then you have side chain. This side chain actually chains and then gives you a new alphabet: A for alanine, C for cysteine, D for aspartic acid, E for glutamic acid, F for phenol. So this is a new alphabet, 20 alphabet you need to know. So these are the all alphabets and amino acids, but the only difference, you have different type of side chain. All amino acid contain carboxylic acid group and amino group, but the only difference, they have different side chain or alkyl group. Okay, so this how protein builds. So amino acids, they join together, they make kind of elimination or condensation reaction. So it's like when two amino acids, they join together, they are kind of getting married together, and when they get married, they actually throw champagne or they throw water. Okay, so they release water and they make a new bond, that is amide bonds and peptide bonds. So then if you have more and more amino acid joining together, then you will have tons of different type of different amide bonds, and it will form polypeptide. Okay, and then when these amino acids, they join together, they can have different secondary structure. They can have alpha helix, they can have beta sheet, they can have loops, and then they also have tertiary structure and quaternary structure. So they have primary structure where you have all the amino acid. Then these amino acid, they can make different type of hydrogen bonds or van der Waals interaction. That way it can form helical form or beta sheet form. And then when these secondary structure, they all stuck together, then they can form tertiary structure. And then if you have different subunit, for example, in hemoglobin you have 4 different subunit, they join together and create the quaternary structure. But in myoglobin you have up to 3-dimensional structure because it has only one subunit. Okay, and then you also have membrane protein. There are two basic type of protein in terms of their solubility. So one is soluble protein. So these are soluble protein, but then we also have membrane protein. They regulate what things can get inside the cell and what things can get out of the cell. So this is one of the membrane protein that's called rhodopsin, that's actually present in our retina. How you actually characterize protein structure and structure? So there are many different techniques. One is protein crystallography. So you crystallize the protein, you actually freeze them, and then you can get the crystal. And then how we take our X-ray, we put ourselves on the X-ray machine, and then we freeze. Okay, we cannot dance or we cannot move, and then we shine the X-ray light, and then in the photographic plate we see the X-ray of the bones or the things we want to see, whether it's broken. So same thing, we freeze protein, and then we put these frozen protein in the sample holder, and then in one side we have X-ray light source that we shine, that goes through the protein, and then it's actually creates some diffraction pattern in the photographic plate. And then from this diffraction pattern, we calculate electron density. And this electron density depends on different amino acid has different electron density. From there, we actually resolve the crystal structure from this electron density. And then we have NMR. So NMR, what it does, so you can put your protein into the magnet, and we applied a magnetic field, and all the nitrogen 15 and carbon 13 and hydrogen, they have different chemical shifts, and we collect all amino acid has their specific chemical shift. Okay, so for example, alanine has different chemical environment, so it has different chemical shift. All 20 amino acids, they have their complete different chemical shift, and when we actually put the protein into NMR, we generate all the chemical sheet, and there is a software that can actually identify all the chemical sheet related to amino acid. That way it can identify all the amino acid present in the NMR, and that way they can actually build. So in mass spec works a little bit differently, so it transfer all the liquid sample to the gas phase, and then you have a mass analyzer that can measure the mass of the protein. So insulin has different mass, cytochrome C or hemoglobin has different mass, so it's actually measured the mass of the protein. And then we have ion detector. It actually measured the abundance, how many molecules present in the sample. And then we have data processing system that's actually gives us the mass spectrum as a histogram. So current proteomics is that you have protein. Okay, so you collected the protein. Let's say you have protein in normal cell or protein in cancer cell. Okay, you extracted all the protein from normal cell and cancer cell, but then you can have lot of different protein. Okay, and protein are big. Okay, so then you use some kind of enzyme. So we usually use trypsin or lysine C enzyme. So we generated peptide, and as we generated lot of peptide from protein, then we need to separate all those protein using chromatography. So chromatography will help us to separate all the peptide. Okay, because if we put all the peptide to the mass spec need time to measure their mass. Okay, so mass spec cannot actually help. It's like let's say if you have a coffee shop, if 100 people goes at once and asking for their coffee, like sometimes if you go to rail station or bus station, if you have tons of people asking for their ticket, the person counterman cannot help them at once. Then the counterman can ask, okay, can you make a line and come one by one so that I know what type of ticket you are asking, so and I can scan your order. Okay, so same concept. When you have tons of protein and you generated peptide, means you are generating more peptide, then you need to separate them. You need to make a line of all the peptide, and then this peptide need to go inside the collision cell in the mass spec, where you break down the peptide into amino acids. Okay, and then you can weigh each and every amino acids. So you will get some fragment of amino acid. You can weigh each and every amino acids because you know the molar mass of each and every amino acid. That way you build what type of protein you have in your sample. So let's say you have normal sample and cancer cell, and in cancer cell, if someone has, let's say, breast cancer, okay, so then they will have more estrogen alpha or the protein related to breast cancer that will be overexpressed. So you will see more of this protein in the cancer cell. But in the normal cell, you will not see that much of protein related to estrogen alpha. So if you have prostate cancer, so you will see all the biomarker protein in the cancer cell. That way you identify all the protein present in normal cell and cancer cell. You can also extend these things for people having diabetes. If you collect, diabetes cell and normal cell, then you can do also the proteomics and all the other disease-related cell. You can collect and identify whether you can identify new biomarker protein, or you can quantify the related biomarker protein already people discovered. Okay, but the traditional proteomics, we use gas to break down the peptide into fragment, means amino acids. Okay, we use gas. Mostly we use nitrogen or helium gas. So this gas, helium gas, interact with the peptide, and then it break down the carbon-nitrogen bonds. So this is carbon and this is nitrogen, and then it's actually produce P and Y ions. Okay, so it's break carbon-nitrogen bonds and produce an P and Y ions. Okay, and we know the mass of all amino acids, and that way we can identify what are the amino acid present in the peptide. And then if we search through the peptide mass, then we can actually identify the protein as well. Okay, so there is a problem in current proteomics. Current proteomics, as we use nitrogen gas, we lost a lot of information. We lost PTM, post-translational modification. So, and that's actually sometimes gas cannot break down the peptide really good. So then if we cannot break down, then we cannot measure the mass of amino acid accurately. So sometimes it can give us false result. Okay, so that's why we try to develop a new method. So during my PhD, so I actually try to develop a new technique. Instead of using gas, so I use UV laser and IR laser. And UV laser to excite the protein, and that excitements actually break down the carbon-carbon bond. So carbon-carbon bond inside the protein, and we use IR. IR actually is a low energy photon. It's actually vibrate the protein. That way we actually can break down the carbon-nitrogen bonds. Okay, and then we can have more fragments. Of course, there is a challenge of data analysis for emerging proteomics, but then we can easily identify all the amino acid present in the protein, and then we can have better investigation, better detection of the protein. So this is the, and also this system doesn't require to use chromatography or enzyme, because some enzyme cannot break down the protein into peptide. Okay, so these can actually, you can, if you have cell and you lysis the cell and you extract all the protein from the cell, and then you can use these emerging proteomics directly putting all the protein in the mass spec. You still need chromatography, but you do not need chromatography. You do not need enzyme enzyme to generate peptide. You still need a chromatography if you want to do high throughput, but you need size exclusion chromatography, not the reverse phase chromatography. We'll be also talking about chromatography tomorrow. So then we also, whatever we eat, okay, so whatever we take as a food, it goes through our digestive system, and then it's started metabolize all the food we actually take. But if people have cancer, people have other diseases, their metabolism is completely different. They produce different type of metabolites. Okay, for example, one you guys maybe know glucolysis. Okay, so glucolysis is a process where you actually metabolize glucose to all the way to ATP. Okay, in normal cell, when you have glucose, that's actually produced kind of 38 ATP in normal cell, but in cancer cell, it's actually it's only produced 2 ATP, because all the ATP are hijacked by cancer cell, because cancer cell needs lot of energy, energy to overexpress and uncontrolled growth. So then it hijacked all the ATP through ATP directly. ATP is supposed to go inside the mitochondria, and then it's supposed to actually generate ATP, but it's hijacked by cancer cell, and then it's actually pyruvate directly go to lactate, and that's why we do not get ATP in cancer cell. So that way you can actually, if you have cancer cell and normal cell, you can identify what's happening in the metabolism level. Okay, same thing if a person having Alzheimer's disease, a person having diabetes. Okay, their metabolism is totally different than the normal person. Okay. So metabolomics has a, like, you have biological metabolomics, you also have environmental metabolism. Environmental metabolism means when you throw something to the environment, okay? Environment also degrade things, and then let's say you throw pesticides, okay? Then these pesticides will be interacting with the sun, also the soil microbe, and then the microbe and the things present in the water, and they will be degrading and they will be creating small molecule from pesticides, that is metabolites. Okay. So any industrial pollution or any things that you actually left to the environment, that's actually also produce different type of metabolites. Also, as I mentioned, so um, once you have these metabolites, you need to extract all the metabolites through different process we'll be discussing, and then you need to separate them. Um, it depends on what type of metabolites you are looking. If you have hydrophobic metabolites, okay, then you can use your column material is hydrophobic, so you can use reverse phase chromatography. But if you have a very polar metabolites, then you can use helic or normal phase chromatography. That way you can separate all the polar. You can also use GC, other technique for separation, but eventually you need to use the mass spectrometry to detect all the metabolites that you extracted. Okay. So there are a lot of mass spectrometry. So once you get the metabolites, you can also fragment the metabolites to know for sure that this is the right and exact structure of the metabolites if you fragment. Okay. And then if you solve those fragments, that way you can do MS scan and MSMS scan. So MSMS means tandem mass spectrometry. So you have metabolites, and then you fragment this metabolites, and you get lot of different peaks that actually help you to build the structure. Okay, so so as I mentioned, that we have genome, so all information in the DNA. Although our training will not be any genomics or anything, and then we have transcriptome, so all the RNA. But our focus is proteome. So proteome is all the protein present in a cell or in an organism. Okay, so that's called proteome, not single protein. All the protein present in a system. So this is our proteomics, and then metabolome means all the small molecule present in a system or organism. That's metabolism. So our focus will be proteome and metabolome. Okay, so this is done for the second lecture, and I will take a couple of questions if you have.
Recording stopped.
Sir, I have.
Yes.
Is you said the emerging proteomic. Is it a type of shotgun proteomic?
No, it's not. Yeah, in one sense, it's little shorter. Means you do not need to separate. You do not need to generate peptide, and you do not need to use any enzyme. So it's called actually top-down proteomics, and the current and traditional proteomics is called bottom-up proteomics. Okay.
Yes, sir. Understood.
But that's what we will be learning. This is just, I'm giving you a broader view that you understand what is proteomics and metabolomics. Because I am thinking that you guys do not have any background in proteomics and metabolomics and mass specs. Because if I think that you guys already know a lot of things, then I cannot teach you well. So my hypothesis
আমার কাছে এটা নতুন ছিল আসলে।
sorry, yeah.
আমার কাছে সবকিছু নতুন আছে আর কি, সবকিছু প্রথম, এজন্য এটা আমার জন্য নতুন হচ্ছে।
আমিও আমার তেমন কিছুই মানে এই বিষয়ে তেমন কিছু তোমাদের ধারণা নেই। আমাকে একেবারে প্রথম থেকে ABC সিটেল থেকে শেখাইতে হবে।
আপনার
স্যার
চিন্তাধারা
এটাই বুঝতে
হ্যালো,
বললাম স্যার আপনার চিন্তাধারাটা অনেক সুন্দর।
ওকে, থ্যাংক ইউ।
স্যার আমার একটা কোশ্চেন ছিল।
হ্যাঁ, বলো।
স্যার পলিসন সেলটা যে এখানে ইউজ করা হয়েছে এটা মানে এই সেলটা কেন ইউজ করা হয়েছে?
ওকে, সো মাসপেকের মধ্যে অনেকগুলো কম্পোনেন্ট থাকে আর কি, ঠিক আছে, যেটা আমরা আসলে এই নেক্সট লেকচারে আমরা দেখবো যে ডিফারেন্ট কম্পোনেন্ট আছে মাসপেকের। ওকে, সো মাসপেকের তো একটা কলিশন সেল থাকে আর কি। একটা কলিশন সেল মানে অনেক বড় মলিকুল যদি হয়, ঠিক আছে। সমস্যা হলো যে তোমার তো অনেক ধরনের আইসোমার থাকতে পারে, তাদের মাস তো সেম, কিন্তু মেবি তাদের স্ট্রাকচার ডিফারেন্ট। Then how you will identify them? So then if you fragment them, that way you can actually identify maybe the methyl group is there, but in other isomer methyl group in other places, okay? But from their mass will be same. So then you need to fragment. So to fragment, তুমি যদি fragment করতে চাও তো প্রথমে তো তোমাকে ট্র্যাপ করতে হবে, তাই না? So we trap the molecules and then we can use laser to fragment, we can use gas to fragment, we can use electron to fragment, okay? So there are many different techniques you can fragment, and once you have fragment, then you can accurately identify the structure. Sometimes from single mass you cannot identify the structure. Because if you have only one mass, if you search PubMed database or any database, it will give you maybe 1,000 compound name, okay? But then if you also have fragment mass, then you can accurately detect a small molecule or also protein. Many protein maybe have similar mass. If you just use the mass of the protein, that way you cannot accurately identify protein. So that's why you need to chop the protein into small pieces, then you can identify accurately.
ওকে থ্যাংক ইউ স্যার।
আমার একটা কোশ্চেন ছিল স্যার।
Yeah, sure.
স্যার আমি যদি কোনো একটা নোবেল পেপটাইডকে মাস প্যাক করতে চাই, তো যখন আমি এটা মাস প্যাক করবো, আমি তো মাস পাবো ইন্ডিভিজুয়াল এমিনো অ্যাসিডের, তো আমি এমিনো অ্যাসিডের অ্যারেঞ্জমেন্টটা কিভাবে বুঝবো, মানে মাস প্যাক থেকে একটা নোবেল?
That actually that will be clarified more in the third lecture, but tomorrow you will get the more clarification. Okay, so যত প্রোটিন মনে করো আমাদের মানে হিউম্যান প্রোটিওম যত আছে, যতটা আমরা অলরেডি আইডেন্টিফাই করতে পারছি সেগুলো এনসিবিআই ডাটাবেজে আছে, ঠিক আছে। and NCBI, let's say in human proteome এখন মানে যে ডাটাবেসটা আছে সেখানে 80,000 প্রোটিন আছে, ঠিক আছে। we can collect all the protein sequence in FASTA file, and then if we use trypsin always cut on arginine and lysine. So whenever you see arginine amino acid এবং lysine amino acid, it cut there. That way it generate peptide. Okay. Then theoretically we can see this where arginine and lysine is there. We can cut and generate peptide from protein. Okay. Then we can measure the mass because we know the mass of each amino acid. And in one peptide, if you have 7 amino acid, you add the mass of 7 amino acid, you will get the mass of peptide. Okay. And then if you have peptide, then you can fragment all the amino acid present and get the mass of the fragment. Okay. That way you can build a theoretical mass list from human proteome. And then you collect the sample from a normal human and the cancer. You can collect the cell from the cancer patient. Okay, let's say you collect the cell from cancer patient. So then you have a cell, and then you need to break down the cell wall. You collect all the protein, but you also need to remove all the DNA, other debris. Okay, we have a protein extraction buffer that helps us to extract all the protein from the cancer cell, and then we we add trypsin on the protein we structure, and then that trypsin actually generate all the peptide, and then we add those peptide by liquid chromatography to separate those peptide, and then once it's actually entered to the mass spec measure the mass of peptide, and then inside the mass spec it actually fragment the peptide into amino acid, and it also measure the mass of amino acid. Okay, then you build a mass list of cancer cell protein or proteome. So then you have experimental mass spec of all the amino acids, peptide and protein in your sample, and then you have human proteome, that's your theoretical, and then you try to match them together.
Okay, sir.
That way you identify what are the protein you found in the cancer cell. But anything actually what you will be learning, okay, step by step. We just started, okay?
Yes, I have one question.
Yeah, sure.
Yes, so how do we employ NMR here? Because as you know, the NMR spectrum becomes very complicated.
Yeah, that's why NMR is not a good technique for proteomics and metabolomics.
So do we
That's why we will not be talking about NMR only, we will be talking about liquid chromatography to separate and mass spectrometer, because then we can do whole experiment high throughput way and online. So that's why NMR is not a technique for proteomics. It's also not a technique for metabolomics. You can use it, but then you'll have tons of complication, overlap, it's very crowded. It will be very difficult to analyze the data. But mass spec is very simple.
So I have a question.
Yeah, sure.
Okay, so we have seen that we can use mass spectroscopy with GCU. So sometimes also we use mass spectroscopy with LC. So my question is it possible to use GCU simultaneously with LC?
Okay. So first thing is that we call it mass spectrometry, not spectroscopy. So when we called it mass spectrometry, not spectroscopy, so that's first I need to clarify. So metry means weighing something. In French word metrics, the unit for weight, okay? So that's why, and we are weighing molecules using mass spectrometer, so that's why we call it mass spectrometry, okay? Spectroscopy is like you have IR spectroscopy, UV spectroscopy, NMR spectroscopy, where you use some kind of light or frequency, and then you see the structural change. You are not weighing anything. You see structural change, and you detect all those structural change because of shifting, or it can be shifting wave number, or it can be shifting chemical shift. So that's why it's called spectroscopy. You are not weighing anything. And mass spectrometry means you are weighing things. So that's why it's mass spectrometry. This word is very, very difficult to pronounce sometimes. Okay. Second is GC means gas chromatography. So GC can only work if you have a volatile component, volatile metabolites, okay? If you have volatile molecules, okay? That's actually evaporate, can transfer to the gas phase very easily, okay? And then you have GC column, which is kind of, separating, it's a silica-based column, and then you, so it's more specifically for gas, like volatile molecules. And for small molecules, okay, small molecules, mass less than 1000 Dalton or gram per mole. Okay, so liquid chromatography, you are using solvent, liquid solvent, mobile phase, and then you have a stationary phase. Okay, and this requirement, even if your sample is not volatile, it can measure volatile, non-volatile, anything. So it's more versatile, but if you are not you are separating by liquid, okay? You are separating based on their polarity, non-polarity. We'll talk about liquid chromatography very details. But in GCMS, that's why petroleum things, petroleum type compounds which are very very volatile, that you can use GCMS. Okay, so you cannot use GCMS for protein or peptide. You can use GCMS for metabolites, but only if your metabolites are easier to volatile, vaporize, then you can. And sometimes if you vaporize, you break down these things, okay? So that's why GCMS is not very suitable, but you can use as a complementary technique for metabolomics and lipidomics, but not ideal technique.
I was asking the question from the science that, sir, I am actually working with the plant extracts, so I have different sort of biopic.
So then plant extract, yeah, you can use GCMS, but for phytochemicals or plant extracts, mostly LCMS would be good.
Okay.
Otherwise, sometimes you also need to derivatorize in GC. Means you are looking for some analytes, but you need to derivatorize. You need to add something with that can bind with your analyte. That way you can use GCMS. So that's also another headache. But in LCMS, you do not need to use derivatorize things.
So does LC can measure both volatile and non-volatile compounds?
Yes.
Oh, okay.
Because LC is liquid chromatography, you do not need vaporize your sample in LC.
Yeah, it was not known to me. I just thought that GC can only measure the volatile compounds and LC can only measure the non-volatile.
No, LC can separate both of them. Yeah. So GC is more appropriate for volatile, and LC is more appropriate for non-volatile and other type of metabolites.
Okay, and thank you, sir. Sir, I have another question. Can I ask?
Okay, sure.
So we have mentioned in the lecture today that protein structures can be altered in different conditions. So if protein structures become altered, so does it mean that its mass also become altered, or is it even same?
Okay, so the mass spectrum will be same, the mass will be same, but you see different charge state distribution. So when protein are unfolded, you will see more charge state. More charge state means protein will get more protonated, because then it will be exposed more to the proton or water, so then it can be more charged. But when protein are folded, okay, so you will see less charge state. So from the charge state distribution, you can identify whether protein is folded or unfolded.
Okay, so sir,
That's what we will be also learning in this class.
Okay, so then in a new disease condition, so we don't know what types of proteins are liable for that disease condition.
Most of your question, you will get most of your question answer the third day. Once you finish, okay?
Okay, thank you, sir. Okay.
So thank you for having all those questions, but you just need to hold it for now. And then once we, complete, then most of the questions, answer you will get by automatic. I do not need to, because even if I answer it you now, you may not understand all those things. I may be wrong, but once you have the full picture, because as I mentioned that I'm assuming that you guys do not have any background in mass spec, so I started like that. Okay, so should we take a little break and then start, or what do you think? Hello?
Sir.
I'm not taking any question right now, because if we take question,
Sir, a little break will be better.
Okay, yeah. So that's
it is
what
the dinner
I'm
time. It is the dinner time, and also we have the prayer session.
Okay, so I think we'll take. So now I think we have 9:28. So we will meet at 9:45. Okay, but you do not need to leave from the meeting. You just need to so there will be nothing